start. And so we can start, and we can start with the rector of the University of Camerino. I leave the room to the rector, which I thank, because I know that he's quite busy, so he found time for giving us a brief hello to all of us. Please, Claudia.
Thank you, Guido. It's a great pleasure for me to bring my wish to this community. I know how is important, how many is important this event. I know how important it is at this moment to study not only how malaria causes disease in the world, but also what methods we can use to combat this disease, which is one of, if I remember what you, Guido, say me some days ago, which is one of the main cause of death. So I believe that events as these are very important because it is very relevant to transfer knowledge, not only from high-level scientists to young scientists, to young researchers, but also to transfer knowledge to the common people, to people living in the community. It's absolutely fundamental because we understand that the society run in a different manner with respect the runners of the scientists run, the science run. So we need to spread the new information, the new results obtained from research, and to share our knowledge with the community. I wish you a very good and interesting and significant workshop. I know that these days for this community is very important, and I want to wish all of you great success in your research in the future.
Thank you very much, Claudia, also to have found the time to be here. It was really nice to have this introduction from you. I leave room to Professor Bruschi, which is the president of Italian Society of Parasitology. Please, Fabrizio.
Thank you, Rector. Good afternoon. On behalf of the board of our society, I want to welcome you to this webinar, which aims to celebrate the World Malaria Day 2022. It is now a tradition since many years to celebrate this day in our universities. I thank all the speakers who accepted to participate and all the people who will attend this remote meeting. Before the COVID-19 pandemic, the term global health was often used with reference to low and middle income countries. If the past 2 years have taught us anything, it is that we are all global health. North or south, microbes and particles connect us all, yet resources are unevenly distributed. This World Malaria Day is a perfect time to recognize that investments in combating diseases that often occur amongst the poorest populations allow countries to build more resilient health systems. The systems can be deployed in response to the next global health emergency. So now I leave the microphone to the president of Italian Malaria Network, Guido Favia, and I wish you a pleasant webinar.
Thank you, Fabrizio. Yes, we would have liked to have held this meeting here in Camerino, meeting in presence, but the pandemic situation driven us to be much more cautious and choosing the remote modality to meet. As Professor Bruschi was saying, it's now many years, 15 years, that the World Malaria Day has been instituted by the World Health Organization. And especially in this year, the general focus was on harnessing innovation to reduce the malaria disease burden and save lives. All of us know that we have no single tool available today to solve the problem of malaria. And in this context, we have invited 3 highly qualified speakers to deliver their talks that will update us about 3 different innovative tools to face malaria. So particularly in my capacity as a director of the Italian Malaria Network and as director of the Department of Bioscience and Veterinary Medicine here at the University of Camerino, I really wish warmly thank all the 3 speakers for their kind acceptance to participate in the event. I'm very happy to see that there are more than 60 people at the moment on the WebEx platform, plus many others are following this event in Facebook, and I'm sure that at the end we will be absolutely satisfied of this event. So thanks again to the speaker, thanks again to all the people participating, and now I leave the room to the chair, Valentina Mangano and Claudia Damiani, that can drive the speaker. Thanks again to all of you.
Thank you very much for the introduction, and I want to join you in thanking the speakers, Roberto Galizi, Craig Bonnington, and Carlota Dobaño, who accepted our invitation. Me and Claudia will chair the session, so basically we will try to facilitate the discussion. So before leaving the word to Claudia to introduce Roberto and his talk, I want just to remind all the participants that you can ask questions to the speakers either by raising your hand. There is a button in the lower part of your screen to do that, and we will check the button in between talks to see if there are questions, and we'll leave you the possibility to do that. Or for those who are following on Facebook, you can type your question into the comment space. So that's it for me, and Claudia, please.
Good morning, everyone, and thank you for joining us at this Malaria Day. I'm giving a brief introduction of Dr. Roberto Galizi. Roberto is lecturer in infectious diseases at Keele University in the United Kingdom, where he is established his research group as part of the Center of Applied Entomology and Parasitology in 2020, after 15 years of research experience at the University of Perugia in Italy and Imperial College London in UK. His research combines molecular and synthetic biology, functional genetics, and genetics engineering towards the development of innovative methods to investigate insect reproduction, as well as technologies for the eradication of pest and vector-borne diseases. In particular, his work has focused on the development of innovative genetic tools, including CRISPR-based gene drive for the eradication of the malaria-transmitting mosquitoes Anopheles gambiae. The title of his talk is Harnessing Novel CRISPR Tools for Malaria Vector Control. So thank you, Roberto. If you're ready, you can start your presentation.
Many thanks, Claudia, and thank you for the nice introduction, and thanks to the organizer, Giorgio, Valentina, all the organizers for the invite. It's a pleasure to be here. I'll try to share my screen. Let's see. And see if everything works. Yes. Okay, I hope you can see the screen. The right
That's
one?
fine.
Yes, it's fine.
Super. Yeah, so as Claudia said, today I put together a few slides to give you an overview of how we've been using, and we are using, and continue to use and develop CRISPR tools to control the malaria vector. I don't need to present myself, as Claudia did a much better job than what I usually do. I'll just start giving you a brief introduction about malaria, which is, as we probably all know, transmitted by Anopheles mosquitoes. They transmit malaria parasites, Plasmodium parasites, from infected individuals to non-infected individuals. And here are a few numbers of the malaria problem today. So there are over 3 billion of people at risk, over 240 millions of cases, and over 600,000 of deaths counted in 2020. The majority of those happen in Africa, and unfortunately happen affect children under the age of 5. It's probably not a surprise that there have been quite a lot more cases in 2020 compared to 2019, which has been recorded as 69,000 more deaths compared to 2019, and is believed that the majority of these additional deaths are linked to the disruptions that have been caused by the pandemic, and therefore disruption in provision of malaria preventions, so drugs and diagnostic and so on. To move to the vector of the disease, today we count over 3,500 species of mosquitoes worldwide. But what we know as well is that only a very few of these species are able to transmit human diseases. And if we consider malaria, the transmission is caused by the majority of the malaria transmission is caused by only 3 or 4 species of mosquitoes. So when we talk about vector control, we're not talking about wiping off all the mosquito species, but we want to target only a few of those that are particularly dangerous. In terms of malaria control methods and challenges, you will hear in next talks about some of the other methods, such as drugs and vaccines. And what I will be focusing on today is the control of malaria vectors. I want to start to highlight some of the challenges that we have today in controlling malaria, both in controlling the vectors as well as the parasites. Which is the first one is the resistance and the efficacy of this control measures. A couple of those ones are the most relevant, probably, are the insecticide, the resistance to insecticides to kill mosquitoes, and resistance to drugs to cure from malaria parasites. The other big problem is been so far is the efficacy of malaria vaccines, and there are also a few other components, obviously, to consider. Consider the African countries are the most affected, so anyway, underdeveloped countries are the most affected from the disease. There is also an additional complication that is derived by the difficulties in distributing treatments and measures to control the disease. So in focusing on the control methods and the vector control methods, we can classify under insecticides or bed nets, which has been highly effective in controlling and reducing malaria cases over the years. That sometimes can be also treated with insecticides. Then another method is the control of breeding sites, and focusing on reducing the number of breeding mosquitoes. And the other method I'm going to talk about today is what we refer as genetic control. So using genetics to control malaria vectors. This is nothing particularly new. A genetic control strategy had been proposed a long time ago. The first form of genetic control strategies relied on the release of sterile insects, which was mostly done through irradiations, with several issues that were related to that, particularly the reduced fitness of these sterile insects that were released in order to mate and sterilize the wildlife females. And over the last probably 20 years or so, the genetic control strategies started to become more and more focused towards the development and the use of genetically modified insects. Three aspects of genetic control technologies is what we consider the highest species specificity. Genetic control strategies rely on the release of genetically modified insects, which will mate with their wild-type siblings effectively, and therefore, by relying on matings, is much more difficult that those control measures would affect species that are not compatible to the ones that are released. Another factor is that can be considered in a sense cost-effective. It's not very difficult to breed insects if you have a good facility. You don't require much cost in terms of food or anything else, and technically also relatively simple to deploy, because effectively what we like to consider is that we ask the mosquitoes to do our job instead of having to treat specific individuals. We release insects that would seek their wild type siblings and do the job for us effectively. Before I go into some of the details about how we're using CRISPR and how we develop genetic control strategies, I would like to just give a brief intro on about the life cycles and genetic diagnosis of mosquitoes, which is not extremely different from humans. Effectively, we get males and females that mate. Only females need a blood feeding, and this is how and why transmit the disease. The blood feeding is necessary to lay eggs, which for Anopheles mosquitoes happens in water. Those will develop through a larva stage to pupae, and then will emerge into adults and start again the cycles. Usually adults, particularly the males, feed on nectars. And the mosquitoes have an XY sex determination. So you would have, as in humans, females have two X chromosomes, males have an X and Y chromosome. The primary signal has been identified. It sits on the Y chromosome, is YOP1, which when the Y chromosome is present in the embryo, triggers the development of all the cascade of signals which cause the splicing of the double sex master gene, and that produces the development of all the male organisms. If you don't have the Y, you will have the female counterpart and the development of the embryo into a female. So nothing really different from humans, apart that they have only 3 pairs of chromosomes. The inheritance of genes or transgenes, which is what we're going to be talking about in a minute, follows Mendelian rules. So if you have a trait, transgene, or a gene in heterozygosis, that has 50% of chances to be inherited in a progeny. This is just, for who may be interested of understanding and knowing how we make transgenic mosquitoes. More or less, the process is similar to many other methods of generating transgenic organisms. We need DNA, which usually is in the form of a plasmid vectors, where we put all the components that we want. Usually we have fluorescent markers that allow us to spot when we were successful in getting our transgenic lines. The methods of insertion of these plasmids into the mosquito genome is through embryo microinjections. So we need some needles linked to a microscope that allows to inject the solution of DNA into the embryos. And then from there, we get, if we're lucky enough, we get our transient individuals, which are then mated to wild type. And in the following progeny, we look for our transgenic mosquitoes under the microscope, and this is how they may look. Before I go forward, you know, the genetic control strategies, you know, do not involve only work in the lab, engineering the lab, and then straight to the field, but is a much more careful and stepwise approach, which involves a lot of different steps, starting from genetic engineering, going through that of characterization in small cages, in larger cages that mimic more closely what happens in the field. And before they go in field testing or eventually interventions, there are additional steps such as community engagement, assessment of risk related to this technologies, regulations, and obtaining the approvals required in order to be able to use your technologies. It's a bit largely very similar to what happens to a new drugs or vaccine, right? Now I'm going to talk a bit more in a bit more details about gene drives, what we refer as gene drives for malaria control. So going back what we said before about Mendelian inheritance, when we have a transgenic mosquito that is heterozygous for a specific trait, you would expect that after a mating with a wild type insect, only half of the progeny will inherit this trait. So what would happen if you release such a transgenic mosquito in the wild is that you don't expect that trait to increase in frequency over time, right? So you don't expect to release 10 mosquitoes and to find after a year hundreds of thousands. Normally, if that trait, the transgenic trait that you put into the mosquito has even a small fitness cost, most likely, even if these transgenic mosquitoes that are released are fertile, you will probably not find anything after a few months. What happens is that if we insert into the mosquito what we call a gene drive element that is capable to bias its own inheritance. What happens is that instead of being inherited, the transgene being inherited by half of the progeny as you would expect normally, you would have the majority of the progeny inheriting this transgene. So in that case, it's expected that over generations and over time, the trait that you have inserted into your transgenic mosquitoes will start spreading into a population and propagating into the population. The speed of propagation depends on the life cycles of the organism where that is found. This is inserted, which is for mosquitoes is very rapid. They usually have about 4 weeks long life cycles, so that the spread of these traits can be very rapid because of this. So gene drives strategies could be, we like to divide them in two different areas, different focus. The first one is population suppression. So and that involves the development of traits that affect the fitness of the mosquitoes themselves. And the expected outcome of releasing such a transgenic mosquitoes would be that over time it would reduce the reproductive capacity and the fitness and the reproductive capacities of the wild type mosquitoes, and therefore reduce the numbers until you suppress the population itself. A second method, which is not something I'm going to be talking about today is using similar strategies to replace wild-type mosquitoes that are capable to transmit the disease with mosquitoes that are refractory to that transmission of diseases, which could be by linking our transgenes to anti-parasites, for example, molecules that have the capacity to kill the parasites once the mosquito gets infected effectively. So we refer to those as population replacement strategies. Both type of strategies effectively rely normally on some couple of specific elements. The first one is an endonuclease. So an endonuclease is an enzyme that is capable to cut DNA at specific sequences, at specific target sites. And this endonuclease normally should be active exclusively in the mosquito germline in order to be able to pass the trait, the transgene, to the next progeny and bias their inheritance, as we will see in a minute. The two strategies that have been working mostly over the last 10-15 years in my former lab are generating meiotic drives or sex ratio distorter. In there, you would have a male mosquito that expressed an endonuclease in the germline. The endonuclease is specifically targeting the X chromosome of the mosquito. If you express this endonuclease in the germline around the meiotic stage, you would impact the sperm that is bearing the X chromosome, allow only Y-bearing sperm to fertilize, and by doing so, the only progeny that these males are able to generate are males. That's why we call it male bias. As you can imagine, that would allow to reduce the number of mosquitoes, of females mosquitoes that bite and transmit malaria. But at the same time, by reducing the number of females, it would also allow you to reduce the size of the populations and therefore to suppress the populations eventually. The second approach is slightly more complex, perhaps, which relies on a endonuclease that cuts the homologous chromosome right in the specific sequence in the place where the endonuclease sits in the other chromosome. If we do this cut in the germline of mosquitoes, what the germline cells like to do naturally is to repair this cut by using the homologous chromosome. If the homologous chromosome carries the egg, so the transgene, at the end of this repair of the broken chromosome, you have two copies of the transgene. So the original copy of the transgene that sits on the other chromosome being copied in the homologous chromosome. This means that starting from an heterozygote trait, you effectively end up in the germline with two copies, such as effectively mimicking homozygote traits in the germline of these mosquitoes. By doing so means that instead of being instead of the transgene being inherited by half of the progeny, the transgene will be inherited by both of the full progeny, because both of the chromosome that can be transmitted carry now the transgene. As we said before, the transgene sits within a specific gene that we can decide whether that gene is responsible for either female fertility or female development. And if we do so, we can combine this increased inheritance of this transgene with the reduction of fertility of females that are that carry this transgene and the disruption that comes with it. Going to the main components that we talked about, so the genetic editor, the endonuclease. We have different types of endonucleases. The original ones that we can call the oldest ones, the oldest versions, were provided by naturally occurring endonucleases, homing endonucleases. They would naturally do what we just said, so cutting and copying themselves into chromosomes, in unicellular organisms, such as in this example is derived from fungi. So the main problem is that in order to take one of these proteins and use it to target a new sequence into the mosquito requires a heavy re-engineering. So it requires a lot of effort in order to take one enzyme that originally cuts a specific sequence in the fungi to recode them and allow us to use them to target a sequence within a female fertility gene or whatsoever. So very complex process. Talents and zinc fingers have facilitated this aspect somehow because they rely on putting together different modules that encode for these endonucleases. And by putting together these different modules, you can quite easily reprogram these talents and zinc fingers endonucleases to target pretty much any DNA sequence you want. Big issue of using those for what we just said, for genetic controls in mosquitoes, is that the modularity of these enzymes makes them very unstable. So you generate a transgenic insect scarring expressing these enzymes. After a few months, these enzymes are not more active and the transgenic line doesn't do anymore what it's supposed to do because you tend to lose some of the modules and so on. Big advantage came when CRISPR came out and offered us a third different genetic editor effectively. We'll have a look in a minute what CRISPR is and where it comes from, but what I wanted to highlight here is that you have two components. So the endonuclease components, the protein, is completely separate from the targeting component of CRISPR, which is effectively only a short sequence of RNA that we call guide RNA, which effectively guides the endonuclease sisters to cut a specific DNA sequence according to the 20 nucleotides sequence that you have on the guide RNA. So that would find its own complementary sequence in the genome, by forming a complex with a Cas9 will allow us to cut that specific sequence in the genome. So obviously that makes it very flexible, easy to redesign. You just need to change this 20 nucleotides and you can easily make a new CRISPR to target any new sequence that you want effectively. CRISPR comes from bacteria. It's nothing that scientists have invented. They found it in bacteria. There are the natural purpose of CRISPR is to offer immunity to bacteria from bacteriophages. So it's a mechanism of immunity the bacteria have evolved to counteract the infections from bacteriophages, which is effectively the bacteria pathogens. There are several different types of CRISPR existing. We tended to take the most simple ones to use for our genetic editing technology. The CRISPR was initially identified in the 87 by a Japanese scientist called Ishino, who accidentally found the clone that an unusual repeat of sequences from bacteria. He didn't know what it was, but later on, through further studies, scientists were able to characterize this CRISPR components, the core CRISPR components, and as you probably know, Jennifer Doudna and Emmanuel Charpentier were able to show the first proof of concept of using CRISPR for genetic editing. They also redesigned the guide RNAs in order to have as a single guide RNA, and they also won the Nobel Prize for this major discovery in 2020. Now let's go to the actual science research that we've been developing around this strategies. First one is I wanted to focus on the sex ratio distorter. and how we developed that by using CRISPR-based enzymes. I'm going to go very quickly on the historical part of the sex ratio distorter. Is something that was attempted quite some time ago. My colleagues at Imperial in 2008 published a paper where they use a natural occurring enzyme called the IPP1, which they were lucky enough that would recognize a sequence that was unique to the X chromosome in Anopheles gambiae, present on the ribosomal DNA sequence, which is a repetitive sequence found exclusively on the X chromosome in Anopheles gambiae. So that is quite unusual, because in many other organisms is not only unique to the X chromosome. But long story short, they were able to use that, express that in the germline, hoping to get a male biased progeny at the end of it. But instead of having that at the end of the process, these transgenic males expressing this enzyme in the germline, instead of generating a male biased progeny, were fully sterile. And the reason for that is because this enzyme was still capable to recognize and cut the sequence that was supposed to cut, but was found to be highly stable, thermostable. So it's an enzyme that lasts for a very long time once it's produced. And because of that, once these males would fertilize and generate the embryos, would also deposit some of this IPPO enzyme into the forming embryo. And that would be active on the X chromosome that was derived from the mother, and therefore kill these embryos that were forming. It took us a bit of time in order to resolve these issues with IPPO1. And as I said before, it took a lot of re-engineering, generating a lot of transgenic lines. And at the end of this process, which was effectively my PhD project, we generated a few lines expressing destabilized form of this IPPO enzyme, so reduced stability, not much activity, unwanted activity in the embryos, and that results in 3 out of over 30 transgenic lines being able to generate a highly male biased progeny and maintain a good fertility as well with it.
Sorry, Roberto.
Yeah.
You have other 5-8 minutes.
That's good.
Okay.
Thank you.
Okay.
Thank you very much. That's perfect. We then moved to developing the same system by using CRISPR. So we effectively expressed the Cas9 and the germline promoter, targeting the same region. And a long story short, we generated 4 lines, and 3 of that were able to generate over 95% of males instead of 50-50 that you would expect in wild type, and also had a very high fertility comparable to wild type. Moving on the CRISPR-based homing gene drives, as we said before, that relies on a transgene, in this case on a CRISPR-Cas, sitting on a gene that is responsible for female fertility or female development. And by using this copy-paste mechanism in the germline, that can be copied across into chromosome. And this is what you expect to happen in a schematic, effectively, when you release such mosquitoes in the wild. You have the spread of this transgene, and once that you have enough of this transgene around the populations, you start getting mosquitoes that are homozygotes for this trait, and females that start to be sterile. Long story short, that works. We have shown that we can bias the inheritance of transgenes. We have shown that we can get the phenotype that we wanted from these mosquitoes. I don't want to go into the details of that, because clearly there is no time. But we also noted that doesn't really work very well. And the reason for that is because when we use CRISPR, when CRISPR cuts into the target size, it cuts the target site, you also have mutations. And sometimes these mutations can generate target genes, so female fertility genes, let's say, that are not cleavable anymore by the CRISPR, but can still produce a functional gene. This is what we call functional resistance that gets selected through populations and stop the gene drive from spreading and suppress your population. A few years after, I just want to highlight what it was, the rapidity since we start using CRISPR in mosquitoes. A couple of years after, we were able to target a highly conserved gene. It would not tolerate mutations in it. And by doing so, we were able to generate a gene drive that was able to spread very rapidly and crush a population in cages over 8 to 12 generations. Problem is, or what many people have started to highlight, was this working too well? Two main reasons. The gene drives can spread very rapidly once released. So there is not much that you can do if you were to release those gene drives into the wild to control them. In addition, this type of gene drives was targeting a highly conserved DNA sequence that could be present also in species that you don't want to target, for example. And therefore, we start to work on some solutions to stop gene drives in case we wanted to. And we went back to the drawing board and to effectively back to the microorganisms that have CRISPR and evolve CRISPR as immunity, as we say in the bacteria. And when we went back, we saw that lots of scientists have discovered, after discovering CRISPR, they also discovered anti-CRISPR proteins, which are proteins that are produced by the bacteriophages that attack the bacteria and use as a mechanism to counteract the CRISPR immunity from bacteria and be able to infect the bacteria effectively. So what we did is try to steal these anti-CRISPR proteins from bacteriophages, encoding them into the mosquitoes in a similar way to what we did for the gene drives that you see in here. We did the same thing, as I said, for the anti-drives. So we have encoded an anti-CRISPR protein under a germline promoter. We generate a transgenic mosquito expressing this anti-CRISPR protein. We did an assay where in the control, we saw the gene drive was able to bias his own inheritance to the majority of the progeny, almost 100% of inheritance. But when we had the mosquitoes that were carrying both gene drives and anti-gene drives, we saw that we could completely stop the biased inheritance of the gene drives, so full inhibition of gene drives activity, and restore to the inheritance of the drive construct to the Mendelian inheritance. We also tested those constructs in what we call cage assays. So we release mosquitoes in small cages and we maintain them over generations. We did some control cages where we only released gene drives, and then we did some testing cages, a couple of testing cages where we released gene drives and anti-drives. So when we release only the gene drives, we saw that the population, as we expected, crashed in few generations. So no mosquitoes anymore, no eggs being produced and no mosquitoes anymore in there. But when we released in the other two cages where we released the anti-drives, we were able to prevent the suppression and effectively rescue the population from suppression. So effectively, our mechanism was working very well. We did some modeling to predict what would happen on a long term after release. We saw that equilibrium between drive and anti-drive and wild type insects depends primarily on their relative thickness. And just to conclude and to give you a couple of information on additional way that we're now trying to use CRISPR for genetic control is by generating CRISPR Biosensor, which rely on the inclusion of switches that we want to use to improve tissue specificity and species specificity of gene drives, for example. I've started a couple of years ago a collaboration with a synthetic biologist who was Professor Jaramillo, originally in Warwick, now moved to Valencia, where effectively we have modified the CRISPR RNA in order to be completely inactive unless this RNA come into contact with a specific RNA sequence, which could be any sort of RNA transcript that could be provided, be coming from endogenous mosquito genes or from pathogens. And in this paper that you can look up, we have tested these rubber switches with a simple in vitro system to sense, for example, an HIV virus transcript, and we saw that this HIV transcript was able to activate this with high specificity this CRISPR RNA construct, as we could measure with this in vitro assay. And what we try now to do is to repurpose this what we call interacting RNA in order to make our CRISPR activated specifically when either in contact with specific pathogens or species-specific transcripts, or also germline-specific transcripts, which would allow us to develop a series of different applications or improve pre-existing ones. And at the moment, we have only a very proof of concept in vitro, where we started testing some of the mosquito RNAs that would be able to activate this CRISPR construct, and we hope to have more data and good news soon. And with this, I would like to wrap up my presentation, and I would like to thank all my ex-colleagues and friends from Imperial College, where I spent over 10 years, and my new host here at Keele, and all my collaborators that are or have participated in a lot of the work that you've seen, as well as the funders, obviously, that are essential in order for us to do research. And I would thank everyone for listening, and happy to take any questions. Hoping not to have taken all the time available.
Okay, thank you, Roberto. I'm introducing Craig. Craig, sorry, you can. Okay, yes. Perfect. Craig Bonnington is a senior technical advisor at Malaria Consortium, a world's leading non-profit organization specializing in the prevention, control, and treatment of malaria and other communicable diseases among vulnerable population. He has worked on malaria project implementation and research for over 10 years with experience in academia, humanitarian and development context, both in Southeast Asian and Sub-Saharan African countries. Craig provides technical oversight to Malaria Consortium seasonal malaria chemoprevention program, focusing on the future of SMC beyond current implementation model, researching drug resistance and its impact on chemoprevention efficacy, seasonality patterns, co-implementation with other intervention, as well as alternative drug regimens and approaches such as seasonal vaccination or monoclonal antibodies. The title of his talk is Seasonal Malaria Chemoprevention, Addressing New Geographies and Drug Resistance. So thank you, Craig. When you're ready, you can start.
Sure, that's great. Thank you very much. So yeah, my presentation is on seasonal malaria chemoprevention, addressing new geographies and drug resistance. And I work as a senior technical advisor at Malaria Consortium, focusing on SMC, but also do other work on drug resistance genomics, and also some work in malaria control in humanitarian contexts. So just to give kind of a bit of background about seasonal malaria chemoprevention, in case there's any of you that are less familiar with this. So SMC, as we call it, is essentially the provision of 3 drugs over the course of between 4 to 5 months in areas across Africa where you have highly seasonal malaria transmission. And so essentially this has been done primarily in West Africa, and there are locations where you have 4 consecutive seasons of drug administration, and in some locations it's 5. And so the drugs that are given are sulfadoxine, pyrimethamine, and amidaquine, and those drugs should be providing about a 28-day period of protection to youngest kids between 3 to 59 months in these sort of highly seasonal locations. So this was something that was approved by the World Health Organization back in 2012. And it was seen as a sort of a new innovative intervention that could use drugs in a chemopreventive way to reduce mortality and incidence in these younger age groups, by maintaining a therapeutic efficacy or therapeutic drug concentrations over that 28-day period. But within this, there were quite a few caveats as to where it was deemed an appropriate intervention, and how it would be monitored as an effective intervention in these locations. So part of it, as I've said, it's restricted to kids for 3 to 9 months, but also as part of the recommendations laid out back in 2012, the WHO recommended that annual SMC rounds comprising 4 monthly SMC cycles would take place only in areas where 60% of clinical malaria cases occur during a maximum of 4 months. And in addition to this, that the therapeutic efficacy of sulfadoxine, pyrimethamine plus amidaquine, these 3 drugs together, is at least 90%. So if you have a lower efficacy threshold, then these drugs should not be used. And so, for example, back during this recommendation, there is a drug resistance marker associated with sulfadoxine resistance, referred to as 540E, which I'll discuss a little bit later. And this was sort of seen as the sentinel drug resistance marker that would indicate as whether sulfadoxine and pyrimethamine in particular would be useful drugs to protect these children for at least a period of 28 days. So as the title of the presentation is thinking about drug resistance in new geographies, what are we thinking about in terms of new geographies? Well, the number of children that received SMC in 2020 in West Africa was around 33.5 million children. And Malaria Consortium, the organization I work for, is the largest single provider of SMC. And so in 2020, we provided drugs to at least 20 million children. And we are now planning to expand that. So traditionally, we've been implementing in Nigeria, Burkina Faso, Chad, and Togo in West Africa. But now, thinking about sort of the criteria in which SMC should be deployed, there is a rethinking as to whether the drug resistance markers are a sensible way to decide whether SMC could be an effective intervention. So they're now basically the World Health Organization has given a green light to lots, not all, but lots of African countries to think about the deployment of SMC in locations where traditionally it hasn't been deployed because of concerns over high levels of drug resistance. But the issue is that despite the fact that SMC has been deployed for quite a long period of time, there isn't really an agreed methodology by which you evaluate the continued effectiveness and the continued efficacy of these drugs. And that has always been an issue for West Africa. But when we think about the expansion of implementing SMC in new locations, that becomes even more of an issue, because we are thinking about deploying this intervention in areas with higher levels of drug resistance. And of course, if you are the director of the malaria program, in most places where you have high levels of, well, you have very high incidence of malaria, falciparum malaria in young children, if you're given a green light to deploy SMC, then that will be taken up. But what we want to do is, Malaria Consortium and the wider SMC community, we want to make sure that as countries think about scaling up this intervention, that we have appropriate research and methodologies to be able to evaluate whether this is an effective intervention in new places, but also how long it's likely to last for. And so as it stands, those methodologies don't exist. So this is what I'm going to discuss a little bit more about as we think about this expansion. So you can see on this map here at the top, that's sort of the traditional range in West Africa, where there are relatively low levels of drug resistance, particularly to sulfadoxine and pyrimethamine, where SMC has now been deployed and is expanding sort of year on year. But then you can see in the map below that this is sort of a band in southern Africa, going along the sort of southern border of Angola, much of Zambia, northern Mozambique, parts of Zimbabwe, and sort of the western edge of Madagascar as well. And then there's also pockets within Uganda, you just miss it on this map, but also in South Sudan, where based on rainfall, there is a good indication that there's highly seasonal incidence of malaria infections in young children in these locations. So again, so the thinking is, well, would SMC be an appropriate intervention in these locations? So thinking about that, coming up to this third map of Africa here on the top right-hand side, this is a paper that had looked at DHPS drug resistance markers. So essentially drug resistance markers and genetic diversity related to sulfadoxine, one of the main drugs used in SMC. And so this is sort of a good indicator as to whether, or was previously believed to be a good indicator as to whether sulfadoxine, pyrimethamine, and amidiaquine would work for SMC. So you can see that in much of West Africa, where SMC has been traditionally deployed, you have high levels of genetic diversity and low levels of drug resistance. Whereas you see in East Africa and Southern Africa, you have a lot of red here indicating highly drug-resistant lineages and low levels of genetic diversity. So you have sort of fixed sulfadoxine resistance, often associated with pyrimethamine resistance as well, meaning that, you know, from traditional thinking that the use of SP would not be particularly effective. And then the final point on this slide is just looking at associations between resistance markers that you find to sulfadoxine. And you can see again that there's an association between two different markers, one called 540E and one called 581G, which are associated with treatment failure if you're using SP, sulfadoxine pyrimethamine. And you can see that East and Southern Africa, there's a strong association between these two markers where you observe complete treatment failure if those drug markers are associated with each other, whereas in West Africa, they're not particularly associated with each other. And then in Northeastern Africa, sort of around Sudan, Ethiopia, Egypt, there is a slight association, but not as much as you would see in East and Southern Africa. So the point is that the profile in these different locations is significantly different. So because of this, what we as Malaria Consortium decided to do beginning of last year was to do basically phase 1 work, not phase 1 trials, but what we're describing as sort of phase 1 research on the use of SPAQ in Mozambique and in Uganda to assess the effectiveness of this intervention. So what we found, and I won't go into this too much because I'd like to focus on what we're doing this year, but what we found was that the deployment using the drugs that are currently used in West Africa is that the deployment was safe, feasible, acceptable to the populations we're working with, and quite surprisingly, we had an extremely high effectiveness measure. Now, this was non-randomized controlled trial that we conducted where we were looking at surveillance data from health facility levels. And what we observed was we had 86% effectiveness in Mozambique and 92% effectiveness in Uganda, where that sort of percentage of incident cases were being prevented through the provision of the drugs. But what was interesting is we found this despite the fact that we had very high levels of drug resistance. So this is one of the slides from Mozambique. So this is looking at PHPS and PHDFR. So these are two drugs associated with self-reduction and permanent immune resistance. I won't go into all the details of this, but these were basically blood spot sample collections that were taken prior to deployment of the intervention and then after the intervention. We don't really observe any differences over time before or after. But what is most notable is that this 540E DHPS marker associated with very high sulfadoxine resistance is extremely high. So you're looking at 65% of these randomly selected samples from symptomatic children have 540E, whereas what we observe from West Africa is extremely low levels. So what this is sort of suggesting is, in terms of effectiveness measures, these markers are not particularly useful, at least at this moment in time, to be able to predict how effective SMC is. And then, sorry, this is another slide looking at resistance markers that we observed to amidiaquine, the other drug. What's interesting is in Mozambique, we see very, very little PFCAT, which is a resistance marker associated with chloroquine, a foraminoglycan drug, which is what amidiaquine is as well. So we don't observe any resistance markers to this, which is interesting. We suggest that amidiaquine is perhaps doing a lot of the work. And then we do have some PFMDR1 multi-drug resistant resistance markers that may be suggestive of some amidiaquine resistance. But again, we do have these very good effectiveness measures. When we look at Uganda, this is essentially similar data from Uganda that's separated by district. I focused on Guido district in Karamoja, where we're implementing SMC at the moment. These are again results from last year. But what we observe again is we have across almost all districts, almost 100% of samples have 540E mutations. So they should be extremely drug resistant to sulfadoxine. We also have a reasonable level of drug resistance to amidiaquine, although it's much, much less than you observe in other locations. But again, really high levels of drug resistance, but really good effectiveness measures. So the question becomes, well, what do we do with this information? If you have really effective deployment of SMC despite drug resistance, do you just deploy it everywhere? Well, what we have decided to do for this year is to essentially evaluate this in a lot more detail for us to be able to discover what exactly is happening. So these are all the components of research that we're doing in Uganda, and we're doing very similar work in Mozambique as well. And we're also just about to begin similar work in South Sudan. But for now, I'd like to just focus on this chemoprevention efficacy component listed here under 2A, because this is essentially a process that we're developing to not just look at effectiveness and sort of symptomatic disease that you're observing at health facility level, but you're also looking at the drug efficacy and the ability of drugs not only to prevent symptomatic disease, but to clear existing sub-patent infection, which in places like Karamoja, particularly in Uganda, is quite likely because transmission is very high, although it does follow this sort of seasonal peak. So you want the drugs to be killing any parasites that are in the kid's blood when you provide the drugs, but you also want those drugs to be able to prevent not just disease, but prevent infection over that 28-day period between doses, so that in the future, your effectiveness measures remain high. You can imagine if you end up in a situation where you have drugs which are preventing disease for 28-day periods, but they're only preventing infection for 21 days, for example. In a couple of years, selection for further resistance may mean that your SMC cycles are only protecting kids for 2 weeks when they should be protecting them for 28 days. So what we've designed is essentially this chemoprevention efficacy study for us to be able to evaluate this. So it's, in terms of sample collection, it's quite a simple process where we collect blood samples after we provide the drugs through directed therapy. We do that day 0, 7, 14, 21, and 28. And we're looking for presence of parasitemia on blood spots, also on malaria slides. We're also conducting questionnaires so that we have weight data, MUAC data, height data. What's really, really important, and this is something that nobody else is doing, is we're also looking at drug levels on day 7 and day 28. So that essentially what we're doing is we're trying to discover whether parasites are surviving, whether those parasites are recrudescent infections or whether they're new infections. But we will also have the drug levels to understand in what environment those parasites are surviving in. And what's really additionally interesting for the work that we're doing in Uganda is we're also exploring not only SPEQ, the traditional drugs that have been used, we're also exploring another drug, dihydroartemisinin and piperequine, which has been used traditionally for mass drug administrations in other locations, but we're also intending to evaluate DP as a drug of choice that could be used in places as an SMC alternative where we find very high levels of drug resistance. So this slide here is just another way of presenting the same data that was on the previous slide, showing you kind of the days that we're taking samples and how we're processing them. But what's really important to sort of drive home with this is that there are established methodologies for the assessment of curative drugs, but there has never really been an appropriate methodology for a robust evaluation of chemoprevention drugs. And that's what we're trying to do so that as SMC is expanded into new locations, we have a sensible and safe approach where we can say, you know, SPEQ or DP or other alternative drugs can be deployed every 28 days in these locations to have high effectiveness measures, but also we know the drugs are efficacious and we know that the effectiveness measures we observe will be maintained for whatever it might be, 2 years, 3 years, 5 years, so that when this data contributes to commitment to change in policy, that there are systems in place so that the drugs are not continuously used and then rendered useless without sort of an alternative drug in place to replace that. And sorry, I'll just maybe go over this slide. But the other point that I make on the slide here is that another important consideration is, particularly for SMC, is the sulfadoxine and pyrimethamine is also used in PTP, the intermittent preventive treatment of malaria and pregnancy. So you also want to make sure that if you're deploying this intervention widely, that you're not threatening other chemopreventive interventions that are using the same or similar drugs. So these are all the things that we're sort of evaluating right now in Uganda, Mozambique, and South Sudan, and we'll also continue to do in other locations as well. So all of this work where we assess efficacy, effectiveness, we're going to be contributing to essentially decision-making framework to help guide countries and governments on the scale-up of SMC in new geographies. So that is particularly applicable to East and Southern Africa, but it's also incredibly important for West Africa as well. And within this, we're hoping that we're going to be able to validate a methodology, a standard protocol that could be committed to global policy for assessing chemoprevention efficacy, so that countries where you do have higher levels of drug resistance, even if you're deploying SMC and you have good effectiveness, you have good efficacy measures that will indicate how long the drugs you're using are likely to last for, so that you can think about changing drugs in the future if that might be necessary. And yeah, I mean, that's the major summary, really. And just that this is hopefully something that we're going to be able to validate, particularly with work in Mozambique by the end of this year, and that a decision-making framework for the wider deployment of SMC and other chemopreventive interventions will be in place within the next year and a half, just so that these things are done safely, responsibly, and any large-scale effects with such a huge number of drugs being deployed over many, many countries is all sort of taken into consideration, and that all the policy decisions that are made are things that will not only work now, but things that are planning for the future. Thank you.
Thank you very much, Craig. That was beautiful. If you are okay with that, I would like to suggest that we discuss the 3 talks together at the end of 3 talks. So perhaps there will be more chance to also discuss how the different tools can be combined. So if that is okay with you, I would now proceed to present Carlota.
Sure.
Thank you very much. Carlota, thank you very much for participating again. Carlota Dobaño is head of the Malaria Immunology Group at the Barcelona Institute for Global Health in Spain, where she coordinates large multi-center studies on naturally and experimentally acquired immunity to malaria. Carlota joined the institute in 2003 at the Manica Health Research Center in Mozambique. Before that, she has conducted research on immune responses to Plasmodium falciparum and malaria vaccine development in Anthony Holder's laboratory at the National Institute of Medical Research in London, in Jenna McWright's lab at the University of Edinburgh, with research work conducted in Malaria Project and Wellcome Trust Center in Blantyre, Malawi, and in Denise Doolan's lab at the Malaria Program of the Naval Medical Research Center in the USA. A main line of our research is the investigation of immunogenicity, mode of action, and correlates of protection of malaria vaccines. And her talk today, which title is Advances in the Understanding of Immune Responses to RTS S, ASO NOR1E, the first malaria vaccines, addresses these very questions. So thank you very much, Carlota. I leave the word to you if you want to share your screen. Thanks.
Okay, thank you. Tell me, please, if you see my full screen.
Yes, we see it now, like in a slides mode.
Okay.
So
let me see, because I have the other one. Let me see if I can take this out, and maybe that would work. Is it now better? Let me try again.
Oh, okay.
Because I have an external, yeah, let me
sure.
see. Is it better now?
Fantastic, great.
Okay, sorry, because I had an extra screen which was taking the, okay, so I don't know how to get rid of this. Hide. Okay, is the audio fine also?
Everything is fine.
Okay, perfect. So thank you very much to the organizers for inviting us to give this talk. So it's very nice that we could have the 3 parts, the vector control and the chemoprophylaxis. And now I'm going to discuss some of the advances in the understanding of immune responses to RTS S. As you know, it's the first malaria vaccine, and mostly giving some illustrations of the work we have been doing in the last years on this. So just as an introduction, so it's not moving. Okay. So it has taken us almost 50 years, depending when we start counting, whether in the late 60s or early 70s, to get this first malaria vaccine. As we all know, because you all work on malaria, it's mostly due because of the complexity of the interaction between the host and the parasite. We are dealing with this very advanced protozoan that has multiple species, multiple strains, multiple stages, and multiple antigens. And within Plasmodium falciparum, which is the one that we work mostly on, we know that there are many antigens that are polymorphic, and some of them are even variants. So this gives the opportunity of evasion strategies to the parasite. So it's then a very challenging task for a vaccine. So, but also from the host perspective, we have multiple immune responses that can act. So both antibodies and cellular effector mechanisms are important. And also all the backgrounds difference in terms of genetics and age that we know are also important for the control of the infection and of the disease. So this results in a very heterogeneous parasitological and clinical presentation in terms of the clinical. But we also have the third component, which is epidemiologically very complex, heterogeneity in terms of transmission, depending on the endemicity, the seasonality. And also in Africa, we know that people are normally exposed to lots of different infections, malaria being one of the main ones, but also other types of exposures like microbiome, etc. And these, we think, are reasons that are maybe underlying why in Africa and mostly children respond worse to infections, and there is this general hyperresponsiveness. So in terms of gaps, we have still to address what is really the best antigen or sets of antigens that are targets for vaccines, because we have to choose among the 5,000 to 6,000 potential targets. We also need to better understand what are the immune mechanisms in nature, but also in candidates of vaccines that really protect, and then we should develop as vaccines. And also how to circumvent this problem that we have, which is a very important issue because it's a target population. So children and African people respond worse to vaccines. So is there any potential way to enhance the immune response to these vaccines in these populations or not? So this has been difficult by not having animal models that are sufficiently predictive. So many vaccines work very well in rodents, but then when they progress to either primates or humans, they don't work as well. So this has been one of the main roadblocks. Also, in terms of innovation, since this seminar is about this as well, so we have had a limited innovation in terms of vaccine technologies, although with COVID, of course, this has been accelerating very much. And with the advent of mRNA vaccines, we have now a new set of potential formulations that we can test for malaria. But in general, we still need a formulation that is able to induce potent, broad, and durable responses. And this is something that has been really stopping us from reaching highly efficacious vaccines. And also, of course, last but not least, we have, with all the poverty-related diseases that affect low and middle-income countries, insufficient investment for both the basic research, but also for the preclinical and clinical development, because this is a very, very expensive operation. So in terms of the leading malaria vaccine candidates, since the elimination efforts started, these the ones that interrupt transmission, so targeting the sexual stages, have been at the forefront of the interest, but there is still no vaccine that is sufficiently advanced in clinical development. Most analysis has been done also to try to develop vaccines that target the blood stage antigens, so trying to mimic naturally acquired immunity that is actually quite efficient in preventing severe disease and death, because with age and with exposure, people tend to become protective, even though the infection still happens, and this is a second strategy. But actually, most of the efforts and the most advanced vaccines are those that target the pre-erythrocytic stages. These are meant to prevent disease and infection, and is the sporozoite antigens that are the main target. So this led us to the first vaccine, which is RTS AS01 or Mosquirix, that is based on a single antigen from malaria. So this is called the circumsporozoite protein. So this is the major antigen that covers the surface of the sporozoite, in an adjuvant from GSK, and this was formulated in a fusion with hepatitis surface antigen and presented as virus-like protein. So the RTS comes from a fragment of the CSP that contains these repeats from the CSP that are highly immunogenic, and a C-terminal part that contains T-cell epitopes, and then fused to these hepatitis surface antigens, together forming these virus-like particles, and then adjuvanted with the ASO1 series, that is a very potent series of antigens. So this was after a long clinical development, first in non-endemic areas and eventually in endemic areas, led to this licensure multi-center phase 3 trial in Africa that was performed between 2009 and 2013. After these years, the European Medicine Agency in 2015 reviewed and gave a positive evaluation. And then when the WHO had to decide, they still wanted to do pilot implementation studies that started in 2019 to address some of the still pending issues that were not clarified through the phase 3 trials in terms of cost effectiveness, in terms of safety and other issues. But eventually, we just recently heard last year that the WHO did the first analysis of the 2 that are planned and gave a positive also evaluation and approval in principle of this vaccine. So we know that the vaccine at the moment is considered to be safe, is highly immunogenic, but the main problem is that it is still modestly efficacious. So it ranges between 28 to 36% in the what we call the children cohort, so children that are 5 to 17 months at the first dose. So here is a schematic representation. So we both tested this in children and in infants, having a booster dose, which is a 4th dose to try to make longer the durability of the response, and then without booster. So having a booster or not having a booster, this is the range, no? 28 to 36. So this is what it is, the situation at the moment. There are second generation vaccines that are in development, but this is what we have at the moment. So the main gaps in knowledge about, among others, but the ones that we are more interested in our group and I will talk about today, is that we still don't know, regardless of these long and expensive trials, we still don't know what is the mode of action of this vaccine, what are the determinants of the immunogenicity and of efficacy, and what are the correlates. And this is some of the reasons why it's also difficult to rationally develop better vaccines and better versions of this vaccine. So with this in mind, we set up in the early times of the phase 3 trial, considering that it could be the last occasion that we had to have a randomized double-blind control trial where we could really compare people receiving the vaccine or not. So we really push as an investigators group the necessity to conduct an immunology study. So really do an effort to cryopreserve extra samples beyond what was already thought for this trial, which was basically for doing basic serology. So we perform additional plasma collection and also additional cellular collection in order to address some of these gaps. So this was what we call the immunology study within the RTS S phase 3 trial that was coordinated in our group with other institutions in Africa and Europe and the States. So the main objectives were to investigate the mechanisms of this vaccine and identify the correlates of protection, but also to understand the effect of 2 very important variables in immunity that are age and malaria transmission intensity on vaccine efficacy and immunogenicity. So the trial had this design. So there were 3 different groups of children being treated. So 2 of them received the RTS S vaccine and 1 comparator vaccine non-related to the vaccine. So the difference between the what we call R3R and R3C is that the first one received 3 doses as a primary schedule of vaccination, and then a 4th dose at month 20, because in previous phase 2 trials, the main problem had been that the efficacy was waning. So this was a test of whether a 4th dose could really improve the benefit of the vaccine, and then compared to another group who didn't receive the RTSS, but a comparator vaccine. And then we collected samples before and after vaccination to start the primary immune response, and also before and after the booster to study the booster effect, and also at late time points, month 32. So it's important to point out that this was spanning in Africa different sites of different malaria endemicity. So in the heat map, you can see those with high transmission or very high, moderate or low. So we had a nice spread. And among the 11 sites that participated in the main trial, a group of 7 sites with different malaria transmission participated in this ancillary immunology study. in which among the 11,000 total that participated in the trial, we collected samples from at least 5,400 volunteers. So just to remind you that there's always been the two age groups, what we call the neonatal one, so the 6 to 12 weeks infants, in which the vaccine efficacy was lower than expected. This was actually the target group, the one that really initially was all designed to protect the most, because they are the most affected by severity. So we had an efficacy of around 30% after 1 year, but then it decayed to 26% after 38 months. And then the main cohort in which everything else has been focused, which is also including the implementation and all the WHO recommendations, is this 5 to 17 months children cohort, in which after 1 year we have 55% efficacy, and after 48 months around 36%. So first of all, I will show you some of the data we have been collecting these years about what is the role of antibodies in protection. So it was already known that antibodies correlated with protection, but we have never had a correlate, and we didn't have much information on the type of antibodies, so both the isotypes or the subclasses of these antibodies. Also about what is the function. Is it mostly neutralizing function, like inhibiting the motility and invasion of the sporozoite, or is it also, or in contrast, a non-neutralizing function, for example, opsonizing phagocytosis or complement fixation? So we were addressing these types of questions that had been not addressed before. So the first thing I'm going to present is just to confirm that the level of antibodies is important, and also the quality of these antibodies. So we performed this study including a subset of 1,000 children from 3 different African sites, presenting the range of low to moderate to high transmission. And what initially we show is that those children that after the 3 first doses of vaccination induce the higher tertile, which are in the gray and medium tertile and blue lines in these survival curves, those that really induce the higher immune response, they were the ones that had the lowest hazard of malaria risk after a year of follow-up. And this was measured by cases of clinical malaria. So this was significant hazard ratio and protective. And this was adjusted by age and by site, that we know are also independently affecting vaccine efficacy. And then beyond that, we also looked at quality, so avidity. That means that those that had antibody responses and were of a higher avidity, so better binding of the antigen, also were better protected, and this was also significantly associated with protection, adjusting by age and site. Next, we ask what happens after this primary immunization when the child is being followed, and then there is a booster or not of the RTS. So this is the blue one is the group that just received the comparator, so no antibodies, and then this is the peak that is observed after the first 3 doses, and then it decays over a course of 20 months. And just before the booster, they are the same, because these up to here are the same. And then those who receive the RTS booster do have an increased benefit. But what we see in this and other studies is that there is like an aberrant pattern, because the levels of antibodies in the booster, normally you would expect to have a higher level than what is a primary immunization. And this doesn't happen with RTS. So this is one of the questions why we are not getting probably better protection, and we are trying to understand why. So, and then this is maintained above the other one. So, but this is still total. This is a very crude estimate. It's just total IgG, but IgG is a mixture of 4 different subclasses. So we went a step forward and analyzed each of the 4 subclasses. So IgG 1, 2, 3, and 4. And as you can see here, the patterns are quite different. So the ones that really mimic the best, the IgG, is the one that has the most contribution. So IgG 1 is the majority of the subclasses, and you can see the pattern resembles very well. But then we see a different pattern with IgG2. So IgG2 increases with the booster, but with the primary immunization, but that doesn't seem to change. So there doesn't seem to be a benefit in the booster, and this is quite intriguing. So we don't really understand why. Then for IgG3, it's similar to IgG1, but as we know, IgG3 is a much shorter-lived subclass, so it really goes down, and then the booster does increase it, but then at lower levels. And then finally, IgG4 also has very low levels initially, and then progressively they increase with age, which is also was a bit surprising. So we are trying to understand what is then the implications of these different patterns. In terms of function, so what does this matter? Because we know that IgG1 and 3 are the typical cytophilic functions that both do complement fixation and lysis and also bind to Fc receptors and then help for opsonizing. So in collaboration with the Burnett James Bison Lab in the Burnett Institute in Australia, we are conducting different functional studies. In this case, this is not from the phase 3, but from previous phase 2 trials in Mozambique for 2 different sites in which we see that there is a strong effect on the vaccination in these functions. So there is complement fixation and there is also binding that is also reflected in opsonic phagocytosis, in this case is with neutrophils. But also, sadly, this activity tends to wane very fast. So this is without any booster. So we see that in the 2 different sites, there is a strong induction, but then the decay is also quite significant, even though there is heterogeneity. So this is really one of the main limitations of this vaccine, and we really don't understand why is the reason. So in trying to go a bit broader, we went beyond what is just ELISA or Luminex, which looks at a few antigens. So looking more on the broad proteome of falciparum with these protein array technologies in collaboration with Antigen Discovery in the States. So these technologies basically can clone and express as many antigens as you want and then print it into slides and then do the sandwich as you would do with an ELISA, and then you can analyze it with bioinformatics tools. So we have done studies on this with thousands of samples from the different sites, and we just basically actually published a paper this last week. But now I'm going to show you some unpublished data that we are still analyzing, in which with this powerful platform, because here we have a lot of antigens up to 1,000, so with IgG responses to 1,000 falciparum antigens, you really can have a very good signature of at the individual level of the level of exposure on the children that is really reflective of the level of transmission in this area. And this is including as many as 6,000 samples. So here we have this Disney projection in 2 dimensions of the data. So this is all the data by different, with the colors depending on the different sites. Each of them is a data point with one of the sites. And that what we see here is that samples tend to group together in terms of vaccine response in relation to the malaria vaccine, to the malaria exposure intensity. So all the responses that are from the higher malaria transmission sites that are shown here, or the lower transmission sites, tend to cluster together. And we think that this, of course, has an impact in the vaccine response. And why is this important? Because we know already from the main efficacy trial. So here you have the vaccine efficacy of all the sites in gradient, no? From the lowest transmission to the high transmission. So you can see that there is some relation in terms of higher vaccine efficacy for those that have lower malaria transmission, and then lower vaccine efficacy for those that have higher malaria vaccine transmission. So with this tool, having the individual level exposure, we can really have a nice signature of exposure and really try to understand better how that does affect the immune response. So, and I just wanted to mention that because the sites with the high transmission have lower vaccine efficacy, that doesn't mean that the vaccine is not good enough, because they have a higher number of cases averted, which is shown here in this graph from the main trial. So basically, even with a vaccine that has lower efficacy, you still have a good benefit because of this trade-off. So this is part of what we are seeing with work ongoing. And then what is also the role of the cellular immunity arm? So much less work has been done, and it's of course very difficult to do these studies in African settings and in children, because you can get really small amounts of blood. So what we tried to do from the beginning was to focus on the peripheral blood mononuclear cell lymphocyte compartment, so looking at CD4 and CD8 T cells, and also looking at innate T cells. So I will show you just one piece of data on some of the work that we are doing with T cells. And this is looking at combination of cytokine biomarkers that can be indicative of function, like Th1, Th2, Th17, etc., or T-regulatory or pro-inflammatory function. So we have been using for many years different kits that contain up to 30 or more cytokines that combined can give you a nice pattern of what is the main bias of the response. So in this particular case, what we did was to get the samples, the cells fresh on site we stimulated them with CSP, so the CSP is the antigen from the RTS S, and then looked at the secretion of the different cytokines. So, and then looking at cases and control of malaria after 3 immunizations. So what we saw is that those cytokines that were mostly Th1 type, in this case interferon gamma, GNCSF, and IL-15, were associated with protection against malaria during the first year of life after the vaccination. In contrast, those children that had a predominantly Th2 signature with IL-5, predominantly IL-5, but also RANTES, were at higher risk of malaria. And this was done with this multi-marker analysis of PLSDA and elastigmat. So this is just one illustration. We are also looking at the intracellular cytokine staining and phenotypic markers to show whether those can give us more mechanistic insights into what these cells do and what happens after a booster dose. So this is still work in progress. So as a summary for up to now, what we know is that in terms of what are the immune responses that in terms of antibodies and cytokines that have been associated with protection, we have like 2 types of responses. So antibodies to CSP after vaccination, including IgG, but also avidity, so both quantity and quality matters, but also the types of IgG subclasses, so IgG1 and IgG3, mostly functions that are according to, associated with complement fixation and opsonization are the ones that are associated with children who are protected after vaccination, and also cytokines of the Th1 type. On the contrary, those children that after vaccination develop these type of responses are associated with malaria risk, and these are actually having IgG2 and IgG4 instead of 1 and 3, but also those children who have higher levels of IgG before vaccination to the CSP are also at worse outcomes. And this is probably because they are children who come from higher exposure. So these antibodies could be actually from maternal origin or as part of their own early life exposure. And also that includes IgM, so children who had IgM, so current infection markers to falciparum pre-vaccination also had high risk of malaria after vaccination. And as I just said, Th2 cytokine bias was also associated with a worse vaccine response. And even though I didn't mention this much, we have also done a series of studies in which we see that, interestingly enough, baseline signatures, so before any child is vaccinated, signatures that can be looked at the transcriptional level or cytokine level and even at the antibody level can also be predictive of protection even before the child is vaccinated. So this has opened like a new field of research in which we are trying to understand whether priming the immune system before vaccination could improve the immunogenicity and circumvent this problem that I mentioned at the beginning of hyperresponsiveness. So this is like work of many years and many different people in the consortium from our lab, but also from the different clinical trial sites who participate, and also from the labs that have participated in some of these analyses. So thanks for your attention.
Thank you very much, Carlota, for your beautiful talk and also for being so perfect with timing. That will actually be helpful to have a nice discussion with the participants and with the other speakers. Would you like to join me on camera, Craig and Roberto, Claudia? And thank you, Roberto. So I would like to first look here at the chat if there are any questions or anyone would like to raise their hand and ask questions actually in person. I can see a question for Carlota in the chat. I may read it. So Professor Bruschi asks is there any relation with IgG4 levels and protection of vaccinated children?
Yeah, thanks. Yes, what we saw in the first study, so the one, I didn't go into all of them, but in one of the studies that we did before looking at the booster effect, is that children who predominantly induce IgG4 or 2, but mostly 2, but also with 4, were associated with also clinical malaria, so meaning more malaria after vaccination. So it was like a marker of risk after the initial vaccination. However, then we looked at what, if you remember, the IgG4 was increasing. So we looked at this same question at the booster dose, but unfortunately this study was smaller, so it was only 50 children from only one site. So we didn't have power to have a significant association, but everything looked like it has changed. So it seemed that the IgG2 was still associated with risk of malaria, but IgG4 may lose this. So it could be something related also to the development of the immune system, or the age, or the fact that children get more exposed, and then IgG4 response changes, could change the function or the phenotype. So in short, yes, for the primary, but not for the booster, it's not clear.
Thank you very much. I can see that Guido wants to ask a question. Please, Guido.
It's for Roberto, which is not technical, Roberto, but when you were rightly addressing the question that we have to go from lab to field, do you have any information about the perception that people, stakeholders or community, village community can have it in using gene editing in general and CRISPR-Cas9 in particular? The microphone, Roberto.
Thanks, Guido. Well, it's a very interesting question, and you know, it's something that we really have to take into account very early on. It's difficult to give a general answer because obviously it's, you know, the perception can be very different. But ultimately, what we really believe is that you know, the final decision effectively should be taken by the you know, the populations affected. And the other thing that I can also add is that, in particular for the eventual use of gene drives, which obviously you know, have the potential to be very effective and clearly much more effective than what we call self-limiting methods, such as the original you know, SITs, such as those ones that have been applied in US, which all, you know, you've probably seen in the news. They also had conflicting perceptions from different sides. You know, what with the gene drives in particular, you know, the other thing you also need to consider is that it's not enough to get a favorable opinion and you know, at the end of the day, an approval from one individual population or even one individual country, because you know, mosquitoes don't have a passport and don't recognize borders. So you know, what we really need is a unified you know, opinion and then a regulatory approval before such a technology can be used in the field effectively.
Thank you, Roberto.
I don't know if there are more questions from the participants. Otherwise, doesn't seem so. I would like to ask a question to Craig. Actually, I have so many, but I'll just start with one. So I really appreciated your talk, and I was wondering if you could tell us a bit more why SMC has proved so great in terms of reaching so many children at risk. You said 30 million about in 2020. And so not only it's an effective measure, but also the uptake and the access that is being reached, it's quite amazing. We cannot say the same for intermittent preventive treatment for pregnancy in women, where also SP used, of course, not in combination with AQ, but Well, we know that there is not such a great uptake from pregnant women during, despite attending antenatal care visits. So I was wondering if you could tell us your view on why IPTP uptake is not as good, and what could be done to improve that based on the lessons learned with SMC.
Thank you. Yeah, so I mean, the great thing about SMC is it's a highly effective intervention in traditional geographies, and it's extremely cost effective as well. So governments are very, very keen for its deployment. I mean, the useful thing of working at Malaria Consortium is we're doing lots of research, but we're also doing lots of direct implementation, and we're working on sort of global policy as well. So we get to see how things work kind of from all sides. And I think one of the reasons that SMC has such high uptake is because basically it's a process of providing drugs at household level. So we have huge numbers of people supporting on implementation. We have very robust pharmacovigilance systems. So if we have any adverse events or serious adverse events, there's a reporting mechanism that's there, that's recorded. You know, if there's an allergy to any of the drugs, that child won't receive the drugs in future. So there's lots of community engagement that sort of places trust in the implementation of the intervention. I think the issue with the deployment of IPTP is that it's health facility based, and that often SP isn't available quite often. So it's maybe not given so regularly. I think there's also maybe, this is conjecture, so we haven't done a study on this, but I think there are provision of treatment of curative drugs in pregnancy is also fraught with complications. It remains the case that a woman in a first trimester of pregnancy cannot, as a first line, be given ACTs. Many countries, it's if quinine and clindamycin are not available, an ACT could be given, or that's not the case and the woman has to be referred. And quinine over a 7-day period often gives tinnitus. There's a danger of ulcerative colitis with clindamycin. So if your first experience as a pregnant woman going to a health facility is that you're given quinine and clindamycin, that there are side effects, trust in health facilities and provision of care is probably reduced as well. And so I think there's probably reluctance on the side of the women that should be receiving appropriate care. There's in addition to an absence of drugs being available. I worked across Africa for many, many years. I've often seen SP that should be given for IPTP is given as a curative drug when there's stockouts of ACTs, for example. So I think all of these things are contributing factors. But the other point to make is we don't really have a good system in place to assess the efficacy of these drugs during pregnancy. So, I mean, we've been deploying SMC at very large scale for a long period of time, but understanding the efficacy of low-level parasitemia over this 28-day period is not fully understood. And I don't think really that there is, particularly in areas in East and Southern Africa where you have high levels of sulfadoxine and pyrimethamine resistance, that there's an appropriate methodology that's previously been used to assess whether SP is actually clearing an existing infection as well as preventing a new infection. And I don't think placental malaria has really been investigated to the level of detail required in those locations where you have high levels of resistance. An alternative that I've been thinking about for some time is if, for example, we were to deploy SMC in places across East and Southern Africa, that chloroquine could potentially be an alternative for IPTP. And with that, then there would have to be lots of community engagement and sort of building trust among mothers for delivery of IPTP, whether it's chloroquine, whether it's SP. But I think there are many, many issues, but I think we really need to assess whether IPTP is working as well as it is, because even if it is preventing disease, carrying a baby to term with low-level parasitemia and drugs which are not able to clear low-level infection is still going to cause problems for that child in the future. So I think, yeah, lots to explore there.
Thank you very much. So that's one very extensive answer to highlight the fact that we need more research into pregnancy-associated malaria and its management, I guess. So I've seen that Giulia Fabri has her hand raised. Giulia, would you like to ask your question, please?
Hi to everyone. I'm a PhD student in Life and Health Sciences here at the University of Camerino. And I have two questions for Professor Galizi. The first one is about the male mating performance of the GM male mosquitoes, since we talked about the disadvantage of the sterile insect technique technologies in terms of a reduced fitness. So I would like to know if some tests were performed in semi-field settings to ascertain the fitness of these GM males. And the second one is about the modeling of the anti-drive gene, because I would like to know if in the modeling some ecological parameters, such as, for example, the overlapping of different generations or migratory flows, they were considered because they can reduce the frequency of this gene inside a natural population. So I would like to know if they were chosen. Thank you.
You're welcome. Yeah, two great questions, Julia. So the first one about the mating, yes, that is important. We do measure these parameters already when we do the small cage experiments, but obviously in those settings, you can still compare versus wild type, but it's a sort of a forced environment, right? This is very small cages where, effectively mosquitoes are forced to mate. So those parameters, as you mentioned as well, are tested with much more accuracy, let's say, when we perform these large cage trials where that mimic more closely what happens in the field, both temperature-wise as well as in a sense with the dispersion. So when we do model the performance of the transgenic lines, among the parameters that we have to put into the model, we also have the mating success of our transgenic insects versus wild type. So those are obviously need to be taken into account both at the very early small cage testing and all the way through the large cage trials and before the eventual field trials and release. So yeah, absolutely, that's very important. With gene drives, you can get around some fitness cost because the fact that they can they have this increased inheritance of these traits. Even if you have a minor fitness cost associated to this construct, you could still have an effective gene drives that, such as the majority of those that we've seen in the presentation. So it's not essential that these transgenic mosquitoes have absolutely fitness comparable to wild type, which most of the case they don't. So, but that in particular for the gene drives, that doesn't affect as much as one would take their effect. And I can say that the ones that we have generated, they more or less degrees, they all have some fitness cost associated. Regarding the modeling of the drive versus under drive, It's the one that I briefly showed that, and we put in the paper that we published last year. No, they didn't take into account of all this ecological and other parameters that you have in a field release. But were much more simple, effectively, and didn't even take into account what you mentioned about the overlapping generations. So those were, let's say, the step one of modeling, effectively, of these interventions. And likewise, what we did initially for gene drives. Say that what you say, modeling what happens in specific ecological settings is very important, because that will, as you said, affect the performance of gene drives as well as the anti-gene drives eventually, if you were to release them. So those modeling are needed before an eventual release, and they need to be tailored effectively to specific settings to have a better idea of what happens, and different population interconnecting, and so on. That is being shown also in some of the modelings that they take in what we say refer as spatial modeling. They take into account these additional parameters. They show that it's a very different performance of same insect being released in different type of settings. Yeah.
Okay, thank you.
No problem.
Thank you very much, Roberto. I'm gonna just check if there are other questions from the participants. I ask Claudia and Guido to help me too with that.
No, I don't see any hands up.
Okay.
Oh, no.
Okay, I read one question from, yes, from Fabrizio. So a general question to Craig: is there a risk to increase drug resistance by the introduction of seasonal chemoprevention in a country?
Yes. Well, the short answer is yes. I mean, the reason that we provide sulfadoxine, pyrimethamine, and amidiaquin is to ensure that you have a longer period of protection, so around 28 days. Amidiaquin and sulfadoxine should both be present, but also you're wanting to expose parasites to those drugs, so that even if they're resistant to some of the drugs, that you're still able to clear a low-level subpatent infection. Now, in East and Southern Africa, we don't really know, I think, is the answer to this question, because we know that we have very high levels of sulfadoxine and pyrimethamine resistance. A lot of those mutations are fixed. So we know there's a problem with those drugs. The question now that I keep wondering about is, what role is amidiaquin playing? And with repeated and continuous use of those drugs together, how quickly will we select for amidiaquin resistance? And I don't have an answer to that question. But we'll be investigating that, and a lot of the data that we'll get from this year will help us to quantify the resistance that currently exists, because there's a few question marks regarding the phenotype of some of these resistance markers. But with that chemoprevention efficacy component of our studies, we'll be able to determine the phenotype of resistance markers that are less well understood. And hopefully we can do that with some of the amidiaquin markers, and that'll help us figure out how quickly resistance will be selected for. We know it will be, we just don't know how quickly.
Thank you very much. May I ask a question to Carlota? There are no others waiting? Okay, so Carlota, could you tell us a bit how do you foresee the future of the CSP vaccine in terms of possibility for improvement of the vaccine itself, and so improvement of the efficacy? And in particular, if you can tell us your view about the possibility of using a different adjuvant. I think there has been some research into the matrix M adjuvant to replace AC-NOR1. or a better epitope. Last year, with this very event, World Malaria Day, we hosted Antonio Lanzavecchia and his work on antibodies recognizing different parts of the CSP, the N-terminal junction, and neutralizing antibody to that epitope should be able to prevent infection of liver cells by sporozoites, if I don't recall wrongly.
You're correct.
And thirdly, what about new technologies to produce the vaccines? mRNA technology, we know now very well, has been employed for COVID-19 vaccines. So can you tell us a bit your view about how these different aspects can contribute to improve the efficacy of a CSP vaccine? Thank you.
Thank you. So you mentioned some of them. So at first level, before even changing the formulation of the vaccine, is to try to deliver it in a different way. So there has been different trials trying to give the what it's called the fractional dose and delay boosting. So instead of the schedule of the 3 doses regular, so having different delays in the vaccination of the booster doses. This seems to be improving in at least in naive US volunteers, so better immune response and better quality. And was translated into African children, and it's been tested. I think we still need to see the whole picture of the results, but so far I think there is no news on a better outcome of that. So that is still work in progress, but it's a possibility. And then the others come in what you were saying in terms of the epitope. So the RTS S contains only the C-term and the non-P repeat, which has always been the immunodominant. And since the discovery of this junctional epitope that had this highly potent neutralizing antibodies, there's been a lot of discussion about whether the addition of this N-terminal region could give an extra benefit. So there is data back and forth. So the epitope is good, but the fact of having this other part of the CSP is not yet proven that it's really going to be better. So it's part of what is now in the testing process. So it could be that it improves the CSP. And then in terms of the other formulations, indeed, the mRNA now has become a very attractive platform because it induces high antibodies. And that's one of the things that we really need for anti-sporozoite vaccines, and also cellular responses. So there are different pharma companies, even BioNTech for sure, and Pfizer are pursuing, but others, I think Moderna too. So the main companies are really trying to develop also malaria vaccine, and CSP is of course the leading candidate. So I'm sure we will hear about this. And also there are other companies with the BLP, different viral-like protein being developed. And then there is the other strategy that is pursued by the Oxford group, in which what they call R21. So this is a different construct that the difference is mostly related to, apart from the matrix M adjuvant, that is different. Also in terms of the quantity of hepatitis B vaccine, surface antigen, I didn't go into detail, but there is the possibility that this combination of the hepatitis B and mostly the ratio of CSP to hepatitis B could be detrimental. That's their working hypothesis. So they had some initial data with promising results, but in phase 2 trials. So this has to be seen in phase 3 trials and longer follow-up. So far these are I would say the leading ones, but there are many groups trying to work on improved CSP formulations and other antigens formulations.
Thank you very much. And I see Craig has written in the chat about the interest of using seasonal malaria chemoprevention combined with RTS S. And that was actually my following question for the both of you. So I would really like to hear a comment. There was this great paper.
New England Journal,
Yes, that showed that if used in combination, the efficacy is much higher than compared to the single intervention. So that that was really great news, and also I think strong
Definitely.
evidence that the vaccine should be implemented in the framework of a national malaria control program, delivering all possible tools of malaria controls. So, but I would really like to hear your comments on what Craig just mentioned.
I think it's very good that these two combined actually perform better. So that's sometimes it happens that with these vaccines, and many of the RTS modifications or like, for example, when they tried prime boost heterologous strategies with adenovirus, they didn't find a better, so that was sadly, they didn't have a better immunogenicity than just the vaccine alone or other combinations. But this is one of the few cases where having, in this case, not a vaccine, but a different intervention like the SMC, there is like really a benefit of having the two. So that was great news. And I'm sure this has also given a lot of impetus to the vaccine beyond what was the realm of vaccines before, but now to the level of malaria control programs and really considering this more seriously than maybe before it was. So.
Thank you. Would you like to add something on that, Craig?
Yeah, sure. Maybe just briefly. I mean, yeah, I've been having a lot of discussions about deployment of RTS S and R21 in combination with SMC. But I think a lot of the conversations initially begin with a scientific lens, thinking about how things can be deployed appropriately to increase efficacy. But then there becomes a question about production of vaccines and what would be most appropriate in the short term. Because obviously kids who are receiving SMC are very well protected. So while RTSS production capacity is a bit lower currently, would it make more sense for those vaccines to protect children which are not in seasonal locations protected by SMC until protection can be increased, or if R21 comes in to replace RTSS? So I think it's interesting because it's, the science is suggesting that kind of co-administration in seasonal areas would be a really, really useful thing to do. But I think ultimately decisions will come back to government budgets and then sort of an ethical consideration while RTSS can't be deployed everywhere. Yeah.
Thank you very much. Guido.
Yes, I have just one more question for Roberto, if can. Just Roberto, just because we are always talking about integrated program, do you think that the use of gene editing is compatible with other measures of intervention? I mean, when you release your mosquitoes, this can be or not affected by, for example, massive use of insecticides, or which is your feeling? Is they are compatible, or there are some concerns about that?
Yeah, I mean, yeah, it's something that we almost now give it for granted, the compatibility with our control programs. Yeah, we do truly believe that all the weapons that are available should be used. Absolutely. I think we can all agree that we're not going to solve such a big problem with just one method of control. So I think combination of all these methods should be the way to go. Regarding the insecticides, yeah, I mean, this is one of the things that obviously need to be considered when before you think about applying a control strategy. As for anything else like vaccines and drugs, you obviously need to consider what else is happening in that place. So obviously it's also relevant to know what you're doing in addition to that. But other than that, yeah, it's absolutely compatible, and there is no reason to think that the genetic control should replace pre-existing vector control strategies, or should not work together with vaccines or drug treatments. I mean, to add to this, I think you've probably seen that there are also studies now in using drugs to prevent transmission, vector transmission of parasites, so to treat mosquitoes with drugs. So, I mean, this is probably one of the most effective examples of combinations of things. But yeah, even with the standards, absolutely, it is the way to go.
Thank you, Roberto.
No problem.
Okay, I do not think there are any other questions. So Professor Bruschi has to leave the meeting a few minutes ago. So I wish to thanks once again the speakers. We are really appreciated your participation today. So really thanks. And this is both from Italian Malaria Network and Italian Society for Parasitology. I wish to thank Valentina and Claudia for chair and for helping me in this organization. And a special thank also to people of our communication office of University of Camerino, which were always very useful in using this platform. Really thank to all of you. I hope next year we will succeed to do it I mean, we can meet each other, but I mean, we hope the situation will be better. And so really thank to all of you. I want to remind to people that the event is recorded. Now we will send it to Valentina to be uploaded to the site of the Italian Society of Parasitology, and also will be on the YouTube channel of the University of Camerino. So this could be useful also for students and for those that told me that they could not participate today but wanted to have the record. So I really appreciate there was something like more than 80 people on WebEx and some tens on Facebook. So I think this day was really appreciated by many people. So thank you to all of you again, and hope to see you very soon. Thank you very much to all.
Thank you.
Thank you.
Thanks.
Bye.
Thank you, Valo.
Thank you.
Ciao a tutti.
Bye.
Take care. Bye bye.